cingulate, in the insula, and also subcortical regions such as the thalamus. While in B, you can see where in these regions where we had gray matter atrophy was specifically associated with degenerating phonoeconomy neurons and Folk cells. And we see that as it would be expected from a regressor derived from the frontal insula, especially the right insula, atrophy here was associated with degenerating cells. Also subcortical regions here, like in the thalamus and in proximity of the stria terminalis, and in particular also anterior regions here, frontal regions such as the anterior cingulate and orbital frontal cortices. And we performed a control analysis to see how specific atrophy within these regions that overlap with the salience network were associated with degenerating phonoeconomy neurons and fork cells. And we've performed a second voxel-wise regression analysis looking at brain regions where atrophy was associated with neighboring neurons, with degeneration in neighboring neurons, pyramidal cells mostly, and found that especially this anterior cluster of atrophy was specifically associated with phonoeconomy neurons and fork cells, possibly suggesting that these are regions where these large-scale projection neurons are projecting their information. In a second step, I wanted to investigate how atrophy within these regions associated with neuron type degeneration were associated with the typical empathy deficits found in this patient. And so, as I said, empathy was assessed through the interpersonal reactivity questionnaire and with the empathic concern subscale, which is a measure of emotional empathy. The higher the subscale, the more empathic you are, the higher the score, more empathic you are. And you can see that there was a negative correlation between empathy functions and gray matter atrophy within these regions of the salience network. So the more atrophy we had in this region, the more empathy was impaired in these patients. And this association was specific. For example, we didn't find any association between empathy and gray matter atrophy of the right occipital cortex, a region which is often normal in this patient population, but also not typically understood as a region important for emotional functions. And also we didn't find any association between, for example, language deficits and gray matter atrophy of these regions belonging to the salience network. And so in the last part of the study, I wanted to put these three pieces together and try to understand, well, what is the relationship between degeneration of specific cell types and large-scale brain system atrophy and empathy deficits found in these patients? Are, for example, from a systems neuroscience perspective, we would think that complex functions such as empathy are not rooted or anchored in specific cell types, but are anchored on large-scale brain systems and that span several brain regions. And so I used here a mediation analysis finding to see how the relationship between phonoeconomy neurons and fork cells degeneration and empathy deficits was mediated through gray matter degeneration of regions belonging to the salience network, and finding out that salience network degeneration fully mediated the link between neuron type degeneration and empathy deficits found in this patient, suggesting that or establishing a link of a chain of influence linking the selective vulnerability of these neuron types to large-scale brain circuit degeneration, to social emotional deficits found in the FTD, and informing us about a possible chain of influence that underlies also normal social emotional functions or social emotional deficits also in other patient populations. So this question then, this research project was important for me, or had an important role in helping me understand the neural system, both at the microcircuit and at the large circuit level that underlies social emotional function. And in the next question, or in the next project that I want to present now, I follow the question of whether we can modulate these systems in an effective way, especially the large-scale brain circuitry that underlies social emotional functions. And in the recent years, a paradigm that has been very successful in targeting social and emotional function is the use of psychedelic substances that include drugs like psilocybin, the major component of magic mushrooms, but also LSD and DMA, which have this intrinsic property of induced altered state of consciousness by docking with high affinity by working as a specific agonist of the 5-HT2A serotonin receptor. And what has been shown with several drugs and with several of these compounds in different clinical populations is that they can elicit rapid and sustained effects in improving affective symptoms and mood disorders. In naive population, after only one session, for example, in this study by my collaborators in Brazil, they were able to show how a psychedelic called Ayahuasca was able to trigger fast and long-lasting reductions in depressive symptom in patients with treatment-resistant major depression. And similar studies have been published at Johns Hopkins, at King's College London, involving LSD, psilocybin, and other similar compounds in similar patient populations. And so in this study I'm going to present, we focused on a psychedelic substance called ayahuasca, which is a psychedelic traditionally brewed by native populations of the Amazonian basin, and which contains two major compounds. First, DMT, which is the substance docking with high affinity to the serotonergic receptor contained in the leaves of Psychotria viridis. And the other are alkaloids, which are contained in this wine, Baristoxis caapi, which act as monoamine oxidase inhibitors and enable so DMT to pass from our digestive system in the brain and to dock to the serotonergic receptor. And we were interested here in assessing the long-term effect of this drug or the subacute effect of this drug on brain network organization. And one way of measuring brain network functional organization is through intrinsic functional connectivity, usually measured with resting state fMRI, where a participant is lying in the scanner for about 10 minutes without any specific task. And we measured these spontaneous fluctuations in the pulse signal, these very slow fluctuations. And the idea is that regions that show a similar pattern of fluctuations, as here, for example, the orange and yellow regions, the orange and yellow time series that are derived from these posterior cingulate and medial prefrontal cortex regions, if they're synchronous and if they co-fluctuate, that they subserve some common function. And it's merely a statistical method to find networks. It doesn't give any information about the directionality of information, but it can help us find regions that are part of a system and that coherently fluctuate across time. And so we use this method, and this method has been successfully used to find, for example, the neural correlates of acute psychedelic experiences, for example, where the patient or the participant is laying in the scanner while the drug is administered. And it has been shown with this method that there are profound changes of this connectivity, particularly within the default mode network, which I mentioned before, but also that there is major connectivity between all brain systems, major large-scale brain systems that can be derived with this method. And so we were more interested in assessing the impact of this drug on the salience network, which has not been studied so much, but also in understanding what are the long-term effects, since these drugs have been shown to have such a profound impact also in the long-term on affective functions. And so about 40 healthy controls were assigned either to the ayahuasca or to the placebo condition. And towards the end of the psychedelic experience, also the altered state of consciousness was assessed through the hallucinogenic rating scale, a questionnaire that assesses questions like how much did your affect change during the experience, or your synesthesia, your feelings of your body, or your motivation, your volition. It's not really, it doesn't have a direction in the meaning that it's good or bad. It's just asking how much your, how much these different dementias are changing compared to your normal condition. And one day before and one day after the session, we acquired resting state fMRI, and we used this data, the baseline data, to seed the salience network here by using a seed in anterior cingulate. And we also seeded the default mode network by seeing a posterior cingulate. And we also see that primary sensory networks, such as the visual and sensory motor network, to to compare these primary sensory networks to higher cognitive networks, since we were expecting that this system would not be affected in the long term by the psychedelic experience. And so we computed change maps between pre and post session for all the networks and for all individuals, and compared then the ayahuasca to the placebo condition, which revealed increased salience network connectivity, particularly within the anterior cingulate and medial prefrontal cortices, while default mode network connectivity was decreased in the posterior cingulate of the default mode network. We also derived measures of between network connectivity by deriving average time courses from the maps that from the large-scale brain network maps that I introduced before, and we assessed whether there were any differences in between network connectivity across the ayahuasca and placebo condition, and we only found increased default mode and salience network connectivity under ayahuasca, and in line with our expectations, primary sensory systems such as the visual network and the sensory motor network didn't show any within, but also any between network connectivity changes across two conditions, suggesting that there is a specific action of ayahuasca on this higher cognitive network that are known to subserve, that contains associative cortices that are known to subserve self-referential and social emotional functions. And as a last piece in the study, we wanted to investigate also what is the relationship between connectivity changes within the salience network and default mode network and the experience altered level of consciousness during the acute psychedelic session, with the basic idea that the stronger the stronger your altered state, your experience of this altered state of consciousness, the stronger also the functional changes should be, the stronger also the functional reorganization of these brain networks in the long term. And so we focused on these three subscales, which were altered under the ayahuasca conditions. So synesthesia, the property of how you feel your body, was markedly changed under the ayahuasca condition. Also, the volition subscale was changed and the affect subscale was changed. And synesthesia changes in synesthesia correlated positively with changes in salience network connectivity, with this connectivity increases, which align very well with the idea that this network plays a specific role in receiving visceral input from our body, from our autonomic nervous system. The findings for volition were not as clear. We found a negative correlation with default mode network connectivity decreases, but there were both in the ayahuasca and the placebo condition suggested that maybe these were non-specific effects, while altered levels of affect during the psychedelic experience correlated positively with increased connectivity of the salience and default mode network. And so this study provided evidence that psychedelic substances such as ayahuasca have also a subacute impact in the long term in these large-scale brain networks that serve higher order cognitive brain networks. And so we found this increases in salience network connectivity, decreases in the default mode network, and increased salience to default mode network connectivity. And that this might relate to changes in interoception, self-referential, and affective functions, which we have seen that are enduring days to weeks after a single session. And as a last piece of this talk, I'm gonna dive now into how this previous project motivated me to develop more fine-grained tools to map these systems. And the basic idea here is that, well, we have this in time, these different functional changes before the session, during an intervention, whether it's psychedelic or something else. And then we might find sustained changes in the functional organization of these networks that might keep this structure for prolonged periods of time, or might also go back to the state prior to the intervention. And so it's inherently asking, okay, can we develop methods that can map these intrinsic dynamic properties of neural systems, including the salience network? And so in the past, what people have mostly been doing is looking at static connectivity, which is just the average correlation across the whole of the duration scanning time of the both activity, for example, in the posterior cingulate and the medial prefrontal cortex. However, there is a growing understanding that neural systems are highly dynamic, and that also with this method, we should be able to measure how brain regions are shifting their connectivity patterns over time. And the basic idea is that if we have here a static connectome, here network A and B, composed by different nodes and positive connectivity in red, negative connectivity in blue, and the strength, like how thick the lines are, reflect the strength of the connections. If instead of looking at the whole scanning time, we look at specific periods of the scanning time, we might see shifts in connectivity. For example, connectivity increases or decreases between specific nodes. Changes in the sign of connectivity between specific nodes, or even more profound changes where nodes change their belongingness to different networks. Here, for example, this network is now part of, this node is now part of network B. And so I applied this framework to study the dynamic functional organization of the insula, which is a key node of the salience network, the main input of visceral information, but which is also known to be very heterogeneous, with more dorsal portions connected to frontal and anterior cingulate and parietal cortices and being involved in cognitive processes, the more ventral portions being more connected to the limbic areas and being involved in emotion, and then the posterior insula being more involved in interoceptive and sensory motor processing. And so I developed a method where if I, for example, derive the ball signal from the left ventral insula, instead of looking at the connectivity for the whole duration of my entire scanning session, I use tapered windows of about 36-second length, which are shifted for the duration of the entire scanning session. And for each of this window, I derive a whole brain connectivity map, showing me the functional connectivity of the insula to the rest of the brain for this specific window. And so I get a window functional connectivity map for each of these windows. And I do this for all, in this study for 218 subjects. And then I vectorize this map. So here, this green row is basically all the voxels from this map here on the top right, and use unsupervised clustering, k-means clustering, to derive modes of dynamic functional connectivity, in which the centroid derived from the clustering approach tell me something about the specific anatomy of these modes, but also the indexes then from k-means clustering tell me how much time participants' insula spend in each of these four modes, and which I can use to derive measures of fractional occupancy, or in other words, how much time did, for example, the left ventral insula spend in mode 1, mode 2, mode 3, and mode 4 in this specific individual. Here, I would just like to give you an idea for how this dynamic maps look like. You can see that there are changes, for example, in connectivity to the visual cortices, patterns of hyper connectivity and then hypoconnectivity. We can see also changes in connectivity here in the frontal areas, for example, here marked changes of connectivity in dorsal cingulate cortex, and then more here orbitofrontal and subgenual cortices. And so this, you can see that the data is very noisy, but this is the dynamic patterns that we are trying to derive with this method. And so I applied this method to derive dynamic connectivity modes for 4 insula seats, the left and right, ventral and dorsal anterior insula. And I invite you only to look at, in panel A, at the first column here, where I'm presenting the 4 modes that were found, for example, in the left ventral insula, where mode 1 was characterized by connectivity of the left ventral insula, in particular to the cingulopercular network, and also lateral frontal and parietal regions. Mode 2 was characterized by hypoconnectivity to visual areas and sensory motor areas. Mode 3 by hyperconnectivity to the same regions. And mode 4 was characterized by a more limbic pattern of connectivity involving the subgenual and orbitofrontal cortices. And basically, similar patterns were found for all my insula seeds, suggesting that these are building blocks or archetypical modes of how the insula is connected to other brain regions in a dynamic way, which is shown here in this matrix. Here you can see that all modes 1 from these 4 seeds were highly similar based on spatial correlation, although there were some differences between the ventral and dorsal domain, particularly on the strength of different mode network anticorrelation, and also how limbic the connectivity was to mode 4 when comparing ventral and dorsal insular modes. And the other information, as I mentioned before, that can be derived from this approach is information about the time. So how much time is my, for example, left ventral insula seed spending in mode 1, mode 2, mode 3, and mode 4? How many transitions between these modes are happening in my left anterior insula? And also how stereotypical are the fractional occupancy patterns? Here I use a measure called Shannon's entropy, where you can see, for example, in this subject is showing a high level of variability in fractional occupancy across the 4 modes. So a high Shannon diversity index, reflecting that the fractional occupancy of the insula in this participant is not very stereotypical, it's transitioning a lot between these different modes. While, for example, this subject, regardless of which insula seed we are analyzing, spends almost all its time in this single opercular mode 1, and characterized so by a low number of transition and by a low number of diversity index. And based on this first analysis where I individuated these modes in 121 healthy controls, I then asked the question in a different sample of how stable are fractional occupancies of insula dynamic connectivity modes across time. And I leveraged for this a longitudinal sample of about 40 healthy controls that were assessed twice with resting state fMRI data in an interval of 6 months. And as you can see, we found very similar modes with this single opercular, visual anticorrelated, visually hypercorrelated, and more like limbic system also in this sample. And through paired t-tests, we didn't find any difference in time spent in these modes across the two scanning time points. Here, the first scan in blue and the second scan, the first scan in red and the second scan in blue. And then I looked whether at the individual level, whether with a more fine-grained approach, I could find some differences across both scanning conditions. And I used a measure called Cousin's similarity, which can be used to assess the similarity between the fractional occupancies profiles. For example, we see here the subject across both scanning conditions spending a comparable amount of time in mode 3. And the same can be said here for subject 41, that although he's transitioning between different modes, the fractional occupancy profiles between the first scan and the second scan are very similar. And most of the subjects were on this high level or on the high spectrum of cause and similarity, suggesting that the fractional occupancy of insula modes is conserved across time, while some subjects showed very marked difference between both scanning time, and we were not really able to, at least there were no relationship to demographical variables on how fractional occupancy of insula modes differed across both scanning conditions. However, by leveraging a different data sample where participants were assessed twice, once with eyes closed and once with eyes open, I was able at least to address some of the factors that might influence why fractional occupancy of dynamic insula modes might differ across two scanning times. And what this study, what this analysis revealed is that under the eyes closed condition, participants spend more time in a mode characterized by visual and primary sensory hyperconnectivity, possibly reflecting the fact that the insula is isolated from these systems due to the eye closure, or even that the participants are undergoing levels of drowsiness. You know, sleeping is a big confounder also in resting state fMRI studies. And yeah, so this project helped me identify, to understand that the insula, although we think of it being a key hub of the salience network, and the salience network such big monolithic static system, is actually pretty dynamic. There were at least 4 states or 4 modes of dynamic insular connectivity, which displayed 4 state characteristics, meaning they could be influenced by external factors like eyes closure and things like that, but also trait characteristic supported by the fact that the occupancy of the different dynamic insular modes were mostly stable in time for most of the participants. And just a short outlook, I'm now focusing my research also on applying dynamic analysis approaches to study the autonomic nervous system. In this study, which I can't share too much, is still at its infancy, we have been looking at how autonomic channels like heart rate, breathing depth, skin conductance, and things like that, how these channels are changing through different emotional states like awe, sadness, amusement, with some very interesting data suggesting that through dimensionality reduction techniques and through data mining methods, we can actually leverage the dynamic properties of this autonomic data to find clusters that overlap very well with the emotional trial where the autonomic data was acquired, suggesting that we can use this data to find emotion-specific autonomic states. And so for the future, what I really would like to do is to have an approach where a multimodal approach where I combine neuroimaging and autonomic activity to see how this continuous interaction between this brain network and the autonomic nervous system is actually is influencing and modulating social and emotional behavior such as empathy, anxiety, compassion, social engagement, both in healthy and clinical population. And I think especially with the advent of interventions such as psychedelics that can induce, that have this very specific pharmacology and induce this marked altered state of consciousness, we have a powerful tool to understand how to modulate these systems and to change the functional organization of the systems to get a better understanding of how they underlie complex social and emotional functions. And with this, I've come to the end of my talk. I'm happy to take any question. I'd like in particular, before that, to thank my K99 mentors, Bill Seeley, Virginia Sturm, and in particular also Manish Sagar, which is the dynamic expert here at Stanford. And I'm very grateful to the Memory and Aging Center. It takes a village to make the studies, and also to my collaborators in Brazil, Draulio and Fernanda, who have introduced me first to psychedelic research. And yeah, thank you very much.
Thank you so much, Dr. Pasquini. And so we're going to move into the Q&A portion, and we have some questions in our chat and our Q&A function. So we have a question from Ariel Keller, one of our graduate students in our lab, asking, thanks for the great talk. What do you make of the difference between mode 2 and mode 3 in terms of connectivity with the visual network? What would the predictions be for linking these modes with behavior?
Yeah, this is a very interesting question. When I first individuated the systems here, mode 3 and mode 2, which are those hypo and hyper connectivity modes to primary sensory areas, I would have actually expected that the occupancy of these modes should be modulated by, for example, eyes closure. And actually what we found out is that this mode 3 basically was not found anymore if we perform global signal regression. So this mode here, this hyper-connected mode, is related to the global signal, which is still hard to understand what it is related to. I mean, there are some data suggesting that it's related to, head movement and respiration artifacts, but there is also a good bunch of data suggesting that the global signal has some important physiological function, and it's maybe coordinating brain networks in time. So I can't say anything about the behavior of the system, but we haven't followed up anymore how it is related to the global signal, but it wasn't present anymore, or only in reduced form when performing global signal regression.
Thank you. And our next question comes from a postdoc in our lab, Shui Zhang, and she asks, as for K means clustering for the dynamic FC of salience network, how do you find the optimal cluster number K?
Yeah, so the data is not as beautiful as here, where you can really see different clusters. It was a little bit more tricky to, let me go, I think I have a slide on this. Oh, here. It was a little bit more tricky to perform, to choose which was the optimal K-mean solution. And I decided to use a pragmatical approach where I would use different types of clustering decomposition based, for example, on 3, 4, 5, and 6, and see whether there were some redundant system that would appear if I increase the K-means, the number of clusters. And so for example, things like silhouette analysis or elbow plot did not reveal any number that was ideal to decompose the data. But what I decided at the end to do was to use an approach where, for example, if one system here, this first mode, was separated in two modes that were overlapping, and which were maybe fragmenting the system in too many major subsystems, I would just keep the lower decomposition. Instead, if I had a mode that was this year, for example, mode 3, that is hyperconnected to sensory, and which here shows also this subgeneral correlation. If in the next step it would separate it in two modes that were more networky, I would keep then this higher mode decomposition. It's a little bit like the question of PCA applied to resting state fMRI, whether if you use a low mode decomposition, you would get like these big monolithic networks, visual default mode, salience network, and so on. Whether if you increase your decomposition to 70 systems or so on, you'll get like a parcellation of the brain. And I used here a pragmatic approach to have at least major systems that we know from the literature, but at the same time not having to fragmentize my K-means or my modes in too many minor subsystems.
Thank you. And then in follow-up, we have another question from Shui, and she asks, and you were using a relative short window length, 36 seconds. Have you explored the effect of window length on the results?
Yeah, I have. I hope I have the slide still somewhere. Oh, right here. Yeah, I explored also by doubling the window length to 72 seconds, and it didn't really affect any of the findings. Like the same modes were apparent when using longer lengths, and also, for example, here you can see that the fractional occupancy between the 36 seconds. Oh, actually, it was 4 times higher now that I see. And the 144 seconds, there were not really big differences in the fractional occupancies across both analysis approaches. So we kept the smaller window length in the hope that it might better map these fast transitions across modes.
Thank you. And we have another question from Jacob Brower, one of our clinical research coordinators. And he says, thank you so much for such a fascinating presentation. His first question is, has there been work looking at impact of ayahuasca on von Economo neurons in particular?
No, because the major problem is that animal models that we usually use lack this von Economo neurons. Like mice don't have them in, I mean, That's not true. Phonoeconomal neurons are found in many mammals, but in this high density within anterior cingulate and in the insula, only in this, in this large brained, highly social mammals like chimps and humans and greater apes, cetaceans, elephants. And these are not the animals that you usually would, that you have in laboratories. And so it's a very fascinating question. There are some labs that are trying to right now differentiate neurons into phonoeconomal neurons. I think this is an ongoing work. In terms of what they express, it has been shown that they express both serotonergic receptors, but also dopaminergic receptors. So it's very unclear to what extent psychedelic substances might have an impact on these neuron types. It is fascinating, though, that if they play a role in social behavior and also in conveying all information from the autonomic system to the rest of the brain, and psychedelics have profound, at least under the acute effects, have profound changes also on how we feel our body, it would be extremely, I would hypothesize that there is a role for these neuron types in conveying the outer state of consciousness found during the psychedelic experience.
Thank you. And then we have a second question from Jacob in follow-up. Deep forms of meditation have also demonstrated similar impact on the salience and DFM network. Do you think we would see an analogous effect with respect to behavior, empathy, social engagement, etc., and or autonomic response?
Intuitively, I would say yes. I don't know exactly how this might mechanistically mediate it, though, because the cool thing about psychedelics is that they have such a clear pharmacology, right? They have this strong agonism action on the serotonergic receptors. And meditation, I don't know, I don't think anybody has a clear idea about how it acts from a molecular perspective on these brain systems. But yeah, many studies shown interesting parallels on also from the neuroimaging side of how specific brain networks are affected. But I think that's an excellent research question.
Thank you. And then I believe we will have Dr. Williams jump in with a few questions as well.
Yes, thanks so much, Dr. Pasquini. It was such an interesting presentation, and so much thought has gone into the different modalities you brought together. So my question is a broad one. Given you find that the psychedelic is increasing salience network connectivity and decreasing default mode connectivity, would you conceptualize the effect in a single session as kind of reorganizing the intrinsic connectivity of the brain, like reorganization across circuits? And then the second part of this question is whether you have thought about implications for other interoceptive effects. So in mental health, we might think of, say, anxiety, because we think of those as involving disruptions in salience and default networks as well.
Yeah, thanks for the great question. This was a study in healthy controls. We were looking also to analyze this data in the depression sample, because I think I didn't mention at the beginning, this trial also recruited patients with treatment-resistant depression, but the numbers were too small, there wasn't enough power. And for example, what people have, or what Carl Harris have shown in the past from King's College London, is actually that the default mode network connectivity seems to be increased post-session in patients. So I don't know exactly how to relate these functional changes in healthy control to changes in clinical population. It might be that the directionality is not the same, since these changes in functional connectivity might reflect very different processes. But I think that it would be very interesting. I completely agree with you that it would be interesting to see what is the relationship of connectivity changes within the salience network and anxiety and other mood disorders or other mood symptoms. Yeah.
Absolutely. So I know it seems like I'm speaking to you from a void. We're still going through some technical challenges to turn the video on, but we'll keep working those out. I'm wondering, we do have clinical specialists in the audience who are working with psychedelics in treatment-resistant depression. So I'll just ask if any of them do want to type any thoughts or questions to follow up on that. And we'd also love to learn more on, even though it's obviously hard to get large samples with these studies. So it'd be good to follow up on that with you as time permits later, just on even what you're finding in the initial subjects. So back to you, Hosner, if you have other questions, and I'll just see if any come up from our clinical specialists.
To note, we did have this live streaming on YouTube and Facebook, and so there were some delays and some missing pieces, but we also have recorded this session, and we will be sure to post that as well and share that with the audience. So we will have that as well.
Wonderful. So I think the comments are just coming through to thank you again for a fascinating presentation. It was a wonderful start to our inaugural seminar series for the Center for Precision Mental Health and Wellness. So thank you for making that possible. And also to wrap up, to thank the audience, and we'll be following up with the next of our seminar series in the near future. And again, take this opportunity to thank those who've made the center possible. So School of Medicine, our department, Psychiatry and Behavioral Sciences, the affiliated organization we are part of, which is VA Palo Alto and the MIREC, and our executive team, which is Dr. Ruth O'Hara, Dr. Max Wintermark, and our executive director, Hosna Omazad. And again, Dr. Pasquini, thank you so much for a wonderful presentation. I'm sure you'll get lots of follow-up questions.
Thanks for having me.
And we'll wrap up now and have a wonderful rest of your week, everyone.
Thank you.